to go ahead and get, thanks, Melissa. So welcome to the fourth lecture of the 2022 Biomedical Research Integrity Summer Lecture Series. My name is Karen Peterson. I'm the Director of the Office of Scientific Career Development and Scientific Ombuds at the Fred Hutchinson Cancer Center. I co-direct the BRI program with my colleague, Malia Fullerton, Professor of Bioethics and Humanities at the University of Washington. We are assisted in our work on the UW side by Melissa Cox of the Department of Bioethics and Humanities, and on the Fred Hutch side by Amber Ismail of the Office of Scientific Career Development. The United States Public Health Service and its research institutions require that all pre- and postdoctoral researchers supported by PHS training grants receive training in the responsible conduct of research. In addition, the Fred Hutch requires all of its pre- and postdoctoral researchers to receive such training. The UW and Fred Hutch have developed this program of lectures and discussion groups to help its researchers meet this training requirement. So just a few bits of housekeeping before we begin. Note that this is a completely virtual program. Attendance credit is tied to the email address that you entered in Zoom, and so therefore it's incredibly important for you to use one email consistently, and please be sure you type it in correctly. If you cannot attend the lecture live, each lecture will be recorded and posted shortly after its live delivery. You'll need to view the lecture and take the corresponding short content quiz before you receive credit for attendance. In addition, for those attending UW-based discussion groups, it's an excellent idea to watch or review the lecture linked to each discussion section before you attend. So we're using the Zoom webinar feature, and that allows attendees to submit questions via the Q&A box that's at the bottom of your viewing screen. And use the Q&A box. If you see a chat box, please don't use that, use the Q&A. So you can submit your question at any time, and you can do that either as an identified participant or anonymously if you prefer. So the Zoom webinar feature has this really great feature where you can upvote questions that you're also interested in having answered by clicking the thumbs up icon that's right next to the question. And so I'll be monitoring that, and Barry will be as well. So please don't forget to complete the post-lecture evaluation that will be sent to you in your registration confirmation email. And this is the primary way in which we can tell that you've attended and engaged with the lecture material. So with the housekeeping taken care of, it is my great pleasure to introduce today's speaker, Dr. Barry Stoddard. Dr. Stoddard is a full professor in the Basic Sciences Division at the Fred Hutch, and he's an expert in protein structure and engineering. He uses cutting-edge laboratory and computational techniques to reveal proteins' 3D forms at the atomic level, which is key to his ultimate goal of modifying natural proteins to treat or cure human disease. His group has made major contributions to the understanding of gene-targeting proteins, from determining their structure to creating variants for use in gene therapy. Dr. Stoddard's work could lead to engineered proteins that are capable of correcting mutations that underlie genetic diseases such as cystic fibrosis, and he's working on proteins that could cure HIV and other chronic infections. Notably, Barry is also the senior editor of the journal Nucleic Acids Research. Barry will be speaking today on themes related to the PHS required topics of peer review and authorship, with the title Good, Bad, and Ugly Authorship and Peer Review Practices Taken from a Journal Editor's Desk. So Barry, I will turn it over to you.
Thanks very much, Karen. I appreciate it. And yeah, let's just go ahead and get going. I've got the Q&A box off to my left here. I'll sort of glance at it from occasion, and let's just do this. So as Karen mentioned, my name is Barry Stoddard, and aside from my daily job of participating in research and running a research laboratory, as she mentioned, I'm one of two senior executive editors of the journal Nucleic Acids Research. And it's from those files and experiences that I'm just going to talk to you today about basically just aspects of the entire publication process. And I know that this particular topic is said to center specifically on reviewership. However, I'm going to be talking about authorship, editorship, and reviewership, as well as broader aspects of whistleblowing when it comes to scientific publication, all of which go together as a seamless whole. You really can't discuss one without discussing all, but we will be concluding with some words about reviewership that should become obvious how they fit into the overall process. So let's just go ahead and start working through this. So these next numbers are taken basically from the past year of my side gig as a senior editor of a journal, May 2021 through April 2022. So the Journal Nucleic Acids Research receives a total of just about 4,100 to 4,200 submissions a year. And I examine, along with my colleague, all of them on a nightly basis, with an eye just to ask a simple question: are they within the scope and criterion of the journal? Do they match the topics that the journal is focused on? And out of about 41 to 4,200 submissions a year, we publish between 12 and 1,300 papers a year. My own personal workload as far as handling papers through the entire peer review process, which means inviting peer reviewers, administering peer review, making an editorial decision, and then adjudicating the subsequent discussion between authors and reviewers to whatever extent necessary, leading to a final editorial decision. I handle about 180 papers a year. So roughly one every other day crosses my desk, and I send it through the entire process of peer review and editorial decision, and all of the communications with the authors that are required to see the process through to the end. So that's the background by which I'm speaking to you today. So relative to those 4,200 annual submissions and about 1,200 publications per year, in any given year, the other thing that a senior editor does is to deal with what we call problem papers that are brought to our attention. And over 12 months, in the past year, and these numbers are very typical from one year to the next, we dealt with 46 total problem papers that were brought to our attention either by direct email or from individual reviewers or authors or readers, as well as anonymous posters on the internet and corresponding anonymous whistleblowers who emailed us directly. So that corresponds to about 1% of the total submissions that we handle in a given year, and about just shy of 4% of the number of papers that we publish. And most of these problem papers are papers that were published in the past, anywhere from a few years ago to up to 20 years ago, as well as some contemporaneous papers. But the majority of problem papers, particularly for category number 1 below, are older papers that have been in the literature, published, discussed, cited, and followed up on for a number of years. Out of those 46 problem papers, about 35 were found over the course of looking at them more closely to actually have problems associated with them that required action, ranging from, first of all, communicating with authors and with readers or anonymous whistleblowers, to then issuing a final action of some sort within the pages of the journal, which range from very simple erratums or core agendums, which just simply correct mistakes, honest mistakes that happen either on the publisher's side or on the author's side, to editorial statements indicating that there are worries about a paper, that people should view the paper with considerable caution, to outright retractions. And a lot of what I'm going to talk about today is going to do with what it is that basically distinguishes between an editorial statement or caution, if you will, versus an outright retraction. And so of those 35 papers that actually contain problems in the past year, they really break down into the following 5 groups. The first one is the worst, for sure, which is inappropriate image and data manipulation. Basically, there's a figure in a paper that looks as if, and probably has been inappropriately manipulated in Photoshop or something of that nature. The second category would be the data hasn't been inappropriately manipulated, but in some way or another, it's substandard to the point that it's looks problematic to a knowledgeable reader. It's questionable in some way. Questions of irreproducibility, questions of the manner in which the data was collected are raised. The third category would be authorship disputes, which almost always occur, not always, but usually occur after publication, potential plagiarism, and one undisclosed conflict of interest. That was this year's category. And these numbers and the ratios will vary from year to year, and some years will have a little bit more of one than another. But the biggest area of concern and difficulty for journal editors and reviewers is category number 1, inappropriate image and data manipulation. So on the next slides, I'm going to walk you through very briefly 3 different examples over the past year and 3 different outcomes, and explain why the outcomes are different. So this is example number 1, taken from a recent submission to the journal. And up top, you see a figure that was part of the submission. And a reviewer writes back to us as part of peer review, and at the end of a fairly lengthy review says, "I am confused by the picture of the SDS gel in panel 1D. It would appear to be shot in color as the standards are in color, but why are the protein bands not blue from Kamasi blue staining? Inspection of the gel picture at a higher magnification worries me a bit. The bands are almost a bit too sharp and clear for a non-reducing SDS gel, in my opinion. Ask for a raw image of this gel." So we look at this carefully, and we magnify it, and we examine it at the editorial level, and you start to see a number of things that are worrisome. And the main thing that we see are clear discontinuities in the background that would indicate splicing from more than one component into this seemingly single gel image. We look at it a little bit further by just doing various types of image analyses. And we start to see not only indications of what look like lane splicing, but we also see some discontinuities that look like they sort of surround individual bands here and there, as if individual components of this gel have actually been pasted into a different background. So this is obviously immediately raises red flags at the level of the journal. And it's brought to us in this case by a reviewer. Sometimes editors themselves will catch this right up front, but we really depend on reviewers to take a very, very close look at the data as presented. And so, in terms of reviewership, your number one responsibility is to really look carefully. If you agree to review a paper, look extremely carefully at the images, at the data, and if you have any concerns or questions at all, raise them so that editors can get in touch with the authors, which is what we did. And in this case, we actually felt at the time that this was so egregious that we should just tell the authors we're going to reject the paper. And we attached the images shown to the left to show what we were seeing. And then we told the authors that we need to see the original data from which the figure panel was constructed, or a full explanation of how this panel came to be, or both. And we informed the authors that if neither are forthcoming to our satisfaction, that we would have to escalate the matter to the institution for further investigation. And this is the second important point, is that when problems are raised, editors and journals can communicate with readers, reviewers, and with authors, but journals and their editors do not have investigatory capabilities that would allow us to proceed further. We don't have access to the original notebooks, electronic or paper or otherwise. We don't have access to individuals in the lab who we can question directly. So we are reliant upon authors to send us raw data, and beyond that, at some point, any problematic paper ends up having to go to an institution for additional investigation, which is the reason why problems that are raised on the internet or in other formats take a long time to resolve, because the back and forth with the institution is the rate limiting step by far. Institutions and departments are notoriously slow at taking care of this bit of business. So in this case, the author came back to us and first of all pointed out that this is actually not a picture of an actual wet gel, but it's actually an electronic gel imaging system, which has been compressed and decompressed in a manner that it produces what are called compression artifacts that make things look discontinuous in the background. But they had, in fact, taken the original gel, which is shown down here at the lower left, and they had spliced out lanes that they decided really did not need to be included in this particular image. So everywhere where they had a lane labeled ambient conditions, they had just taken those lanes out because in the end it wasn't necessary for this figure. And then they stitched together the gel to make this composite. So this is the original raw data that they used to construct this composite image here. And then here is just an explanation of the lanes they stitched out. So this rises to the level of certainly poor authorship practice. You can never ever stitch together a gel or cellular microscopy fields or anything from separate components to create a composite and not clearly outline and explain the nature of the composite that was made and show exactly where the discontinuities are. But there was no intent to deceive here. The individual lanes that they're showing are faithful reproductions of the lanes that they intended to show. They simply failed to describe the manner in which the figure was put together in the original submission. So we actually reversed the rejection, had them resubmit the paper with this figure correctly annotated and described, and then the paper is still going on right now. The key point I want you to take away is the reason this had a relatively happy ending is because the authors were able to produce the original raw, uncompressed, untouched image from which the figure was made. And that is the absolute key that I'm going to emphasize over and over again. That was what they were able to do that allowed the train to stay on the tracks. So here's the second example, and this one I can actually show the full authorship and the name of the paper because this is all present in the public domain online. This is a resolved, finished case. So in this case, somebody comments on an internet posting service called PubPeer, the online journal club, that they see what appear to be replications of, in this case, actin loading controls for what should be completely separate experiments, okay? So this is originally posted on PubPeer. Now, we didn't see this because as a journal, we do not proactively monitor PubPeer and Imgur and other online posting services where these types of images and questions are posted. We rely on anyone, anonymous or not, emailing us. And as soon as we get an email, then it's an active thing that we have to investigate every single time. So a number of other people post on other aspects of the same paper, actin panel, actual experimental bands, not loading controls, but actual experimental bands that look to be suspicious in terms of how similar they are and so forth. And then finally, we get the email corresponding to all this. So we get an email from an anonymous person who goes by the pseudonym Claire Francis, someone who does a lot of this, basically asking us about the exact same paper, figure 1A. And so this email is pointing out actin loading controls for two separate figures. And it's basically stating those are pretty clearly the exact same loading controls reported for two separate experiments. So at this point, we go to the authors and we ask them what's going on. And to make a long story short, in the end, we end up issuing an editorial note of concern. So we state that we were first alerted in 2013 that the blots depicted in several figures show unusual levels of similarity. We looked at the time, and with the tools that we had at our disposal and the criterion that we used in 2013, we weren't able to, with 100% conclusiveness, determine that there were, in fact, inappropriate manipulations of the figures. We thought they looked funky, but we have a high standard for coming to a conclusion that authors have actually misbehaved. So we just left it at that. Then the same allegations were raised again in 2021. And at that point, we have much better tools and much tighter criterion for inappropriate manipulation. We write to the authors. We tell them we're looking at this again. The authors do not have any of the original data available. So then we go to the author's institution and ask them to perform an internal investigation to try to find out what happens. And so this is just the summary of the allegations for figures 1A, 2C, 3A, 3B, things that are more similar than one would expect by random chance, things that look like they've been copied and reported as being different experiments that look like they're the same. And in the end, we get an institutional report back. And the institutional report says, in light of the site visit, the study of the work, and the rules that scientific journals had at the time of publication of the article, of the repetition of the experiments under investigation, the Commission unanimously believes there has been no alteration of the figures or published results, as amply demonstrated by the repetition of the experiments with the same reagents and protocols, and by how the scientific community, based on the published data in the NAR paper under investigation, were able to progress the study without detecting any conflicting data. So the institution basically says, no, we don't have the original raw data, but the study seems solid, and everybody, and both the lab and others have been able to reproduce all of the results. So we don't have no reason to believe that anything has been unduly altered in an inappropriate manner. So you can see where that puts us as editors, because things look not quite kosher, but the institution, who is the formal investigative body always, comes back and says they think it's fine. So we go ahead and we issue just an editorial note that is basically a caution. And so we've faithfully stated exactly what all the things were that were brought up and questioned. We provide the conclusion of the institutional report, and then we say, in the absence of the original data, we cannot conclusively confirm whether or not the results and conclusions presented in the article are affected. We therefore advise readers to examine the details of the study with care. So we're stopping short of a retraction, but we're putting sort of a caution on this paper. Let's look at one more. So here's a paper where, once again, people are commenting on PubPeer, and then we received emails pointing out bands that should be completely separate experimental samples that look really, really similar, okay? And this is just one little panel out of a whole bunch of figures for this paper that have these irregularities where things just look more similar than really you would expect by random chance. So in this case, we do the same thing. We go to the authors, we ask them for the raw data, and again, the authors have no raw data. They cannot send us back the original gels, the original blots, the original scans. And they just simply say the same thing. We didn't do anything wrong, and even if these figures look funky, all the stuff is reproducible, and there's no reason to think that the paper is incorrect. So we go again to the institution, again, the final arbiter of this matter, and they investigate. And they say as follows: the original data and images were mislaid sometime before 2017, no longer available for inspection analysis. The only images that are available are in PDF form, not of sufficient quality for conclusive analysis. So it's impossible to determine whether the original images were manipulated unethically, as has been alleged, or if they are correct. The original authors maintained throughout the investigation that the images were not manipulated in any way. And they've repeated experiments and generated additional data confirming the results. But they then say, since the raw data and images are no longer available for inspection and the college can't confirm categorically whether the images have been altered, the college's view is that although the results themselves are not in question, the paper should be retracted. So this is the exact same situation as the previous paper, but the institution is coming to a different conclusion because the investigatory body is populated by people who have different standards for author behavior. And in this case, this investigatory body at Imperial College in London felt strongly that when questions were raised and the authors could not produce the raw data, that throws things into enough of a question that the paper should be retracted. And so it was, okay? And we provide a retraction, we just faithfully state exactly what happened, and we state the conclusions of the investigatory body. So hopefully, one of the things that you have suddenly realized is that there's some subjectivity in sort of like the final outcome of these things, depending on two things, really two variables dictate. The first is just the investigatory body, the author's institution and investigators there, and what they conclude and what their standards are. And the second is whether or not the authors can produce the original raw data. So there's a question in Q&A from Catherine. Do authors have the ability to retract a paper if an editorial note is made? Or is that almost a permanent record that they had compromised data according to the editors? So of course, authors can always come to us and say, this paper was we have discovered our paper is fundamentally flawed and we'd like to retract it. In which case the paper still exists on our publication platform and on the internet, but with a big retraction notice associated with it. But in most cases, authors will simply, in the end, live with the editorial note of concern if that's as far as we go. And the editorial note of concern is exactly what it means. It's just a warning, a statement of concern to view the paper with some extra caution, because questions have been raised about the nature in which the data and the figures have been assembled. All right, so finally, here's one very recently, just came in 2 weeks ago. And it's actually concerning a paper by my colleague in the senior editorship. And the only reason I point this out is that somebody wrote in and said, I got a bunch of questions. And the first one is, the authors state that mutations generated by site-directed mutagenesis were confirmed by DNA sequencing, but no sequence data are made available to validate this statement. So in other words, they're complaining, not that they see something wrong necessarily in the paper, but you didn't upload your sequencing traces from quick change. When you change that aspartate to a tryptophan, you didn't, and I don't know about the rest of you, but this is new to me. I, have made a lot of mutants and a lot of proteins, and I've never actually provided as supplementary data the sequencing runs as supplementary data, so people could see that I wasn't lying about the output of the sequencing. But that's not to say I don't have that data stored on a hard drive for a paper. And the rest of the questions go along the same line. You didn't show us this, you didn't show us that. No questions about things that look wrong in the figures of the data, but questions about why didn't you provide this raw data? Why didn't you provide that raw data? To which the author goes ahead and uploads every single last piece of raw data, from sequencing traces on down to micrographs and everything else, into a publicly available database, and then posts a note on PubPeer and at the journal saying, questions have been raised, here's all the raw data, every single last bit of it, and the problem goes away. All right, so this leads me then to best authorship practices, many of which really are about not only best authorship practices, but protecting yourself as a scientific researcher and an author. And at this point, here are some of my conclusions. First of all, there is absolutely no statute of limitations for having your published writing, figures, or data questioned publicly. Some of the papers that we receive emails about were published over 20 years ago. A lot of them right sort of at the advent of Photoshop, when, not surprisingly, people started doing things that they shouldn't have. So no matter how old a paper is, if somebody wants to start blogging about it, posting about it, emailing editors and journals about it, questioning it publicly or privately, they can and they will. And editors must follow up. There's no such thing as saying that paper was published 10 or 20 years ago, so time has run out. That's just, it doesn't exist. There is no effective way for an author to argue with the internet. Okay, if somebody posts about you on PubPeer and you try to argue, you're just going to be in for a world of hurt. So that there's arguing points of the dog ate my data, or we've reproduced this experiment a ton of times and it always comes out the same, or this paper's been cited 1,000 times, people have followed up in all sorts of ways and nobody's ever questioned it before. None of those work, because people will always return to the simple question, that's fine, but what about figure 1D? What about that figure in the paper exactly as it was presented? You have yet to tell us why we shouldn't believe that it's inappropriately manipulated. So you can come up with every possible argument that the paper's fine, but people will still return to the item at the question of what about figure such and such that seems like it's inappropriately manipulated. There's no such thing as protection from double indemnity. And for anybody who isn't familiar with American jurisprudence, double indemnity in our judicial system means that once you've been acquitted for a crime that you were accused of, you can never be put on trial again. Okay, but that doesn't exist in science. You can be cleared of misdoing such as it was in 2013 or 2010 or 2017. And if somebody raises questions again in 2021, the paper and the figures in the paper can be examined again with entirely new and much more sensitive tools for inappropriate data manipulation. Manipulated act and loading controls are just as deadly to an author's reputation in terms of having their work questioned on the internet as a manipulated main figure panel. So anything can be questioned, including, as you just saw, your quick change sequencing. So this is the key point, that if you take only one thing away from the entire talk today, this is the point to take away. Your best and only protection and the best possible authorship protocol and policy is to always have your original, uncompressed, unaltered, unimpeachable raw data and raw images saved, archived, and available for everything from DNA sequencing for a point mutant right on up to cellular micrographs, microscopy images, gels, blots, and everything else. If you have that data available and in this day and age archived at a publicly available database, corresponding to every single data, image, and figure that you put into a paper, you're going to be fine. If you do not have those things, you're playing with fire. Okay, so most journals now are actually flat out requiring deposition of all raw data or providing it as supplementary data that should live with the journal. And so for run-of-the-mill biochemical data, gels and whatnot, there's a variety of services available. The Harvard Dataverse, Dryad Depository, Zenodo, you name it. So again, I just want to sort of conclude this part of the talk by just stating again that in this day and age, with the combination of so much data being digital in its final form, and with the internet and investigators who use the internet being extremely active, not only in examining figures, but posting about figures and then talking about figures, it is absolutely essential as an author to maintain the presence and the retrievability of your original, uncompressed, unaltered, unimpeachable raw data. So let's see here, we got a couple questions. In case of raw data not being available, if the raw data is obtained again, would that still be acceptable? So in other words, somebody questions your blot, and so you go back and redo the experiment, maybe even with the same exact reagents, and you get the same basic result. Is that acceptable? You would think so, but the answer is no, because the question is not whether or not the result is correct. The question is whether the image or the data that you presented in your paper was inappropriately manipulated. It may have been inappropriately manipulated, but still accurately reflect the basic result. But if it's inappropriately manipulated, you'll never get out from under that argument. So, and we do get this all the time. We will always hear from authors saying, not surprisingly, yeah, those blots from 20 years ago, we don't have them anymore, but we can go back and redo the experiment. In fact, we already have, and here's a different version of the same experiment. And that's great. We're glad that the experiment can be reproduced, but it doesn't address the question that everybody just hammers on PubPeer, which is, what about the original figure that you published? What are best practices for engaging with public criticism, i.e. on the internet, without resorting to the dog ate my data style defense? Is it best practice to just point to the original raw data and say, have at it? Yes, Tim, that is exactly the best practice. If you don't have the raw data, and I just say this as someone who has watched these dramas play out in real time on the internet, and people are just not satisfied. If you don't produce your original raw image, similar to that author in paper number 1. Remember that figure that I showed you, the very first example? It looked pretty awful, right? I mean, but it turned out it looked awful largely just because of compression artifacts coupled with inappropriate splicing without an intent to deceive. If they had not had the raw data and that paper had actually gotten published with that figure, they would have gotten savaged. They would have gotten savaged, but because they had the raw data and could show exactly what they did, that was sufficient. Are there some forms of data that should not be made available or should not be made available in raw form? Sensitive data collected, and the answer to that is absolutely yes. So there are all sorts of regulations, particularly relevant to human clinical data or of any form. And that, of course, then brings up an entire additional layer of complexity if somebody's data is questioned in some way. At that point, basically the entire process has to be run through a secure mechanism to preserve security and anonymity and so on. All right, so let's move forward just a little bit. Hopefully I've pounded this in enough that, again, from a journal's perspective, we see all sorts of problems. I gave you that list of 5 at the beginning, but data figures that look inappropriately manipulated are at the absolute top of the list for any journal. And in the end, again, raw data is the original raw data is the only thing that you can really count on. So how does this all sort of go down in terms of like if a reviewer thinks that he or she has spotted something questionable or sketchy, if an editor has received an email from a reviewer or from a reader or from an anonymous whistleblower, what are the rules? Are there rules that guide how this process happens? And the answer to that is yes, there are rules, and the rules are actually available on the internet through COPE. COPE is the Committee on Publication Ethics, made up of a large number of scientists, journal editors, and publishers who basically have produced the protocols that editors, publishers, and those involved in public education ethics should utilize in order to promote high standards of research integrity during the publishing process and subsequent questioning of the publishing process. So I strongly encourage all of you at some point in your training time to take the time over a pot of coffee or your favorite libation to go to publicationethics.org and just read up what COPE has to say. And one of the things that COPE has is it provides online guidance, searchable, regarding various types of scenarios, intellectual property, peer review processes, post-publication discussions and corrections, allegations of misconduct, and so on. So for almost any scenario, you can search and find a topic that will allow you to enter the flowcharts of what to do if. So in this case, what to do if you suspect peer review manipulation, or what to do if you suspect image manipulation in a published article. And those will lead to these sorts of documents that as an editor, you can sort of follow. So I won't take time to read every single bubble, but you can sort of get the basic idea by just sort of looking at it broadly. Somebody, either a reviewer or reader, expresses suspicion of fabricated data. In the best of all possible worlds, the reviewer spotting it during peer review. In the worst possible scenario, it's an already published paper, already cited paper, and a reader brings it to an editor's and a journal's attention. But in any case, the flow chart is going to be the same. You're going to go get a second opinion to see if you think you see it as well. You're going to assemble what the question is, any evidence that you have. And you're going to contact the author. This is going to be the first point of contact, and you're going to express concerns. The author is going to reply or not. The reply is either going to be satisfactory, as was in the case with the very first scenario that I showed you, or it's going to be unsatisfactory. maybe the author will admit guilt, although I've never actually seen that happen. And at some point, you're going to ask for the raw data, the lab notebooks, and maybe you will get to see that raw data. Maybe you won't. And at that point, you're going to end up with the institution and or a corresponding regulatory body if the problem is egregious enough. Typically, what happens is editors and journals work with the corresponding institution and department where the authors are located. And then if that group finds a problem, then generally a regulatory body, often the Office of Research Affairs at NIH, will get involved looking more deeply. So let's see here. So does this mean that a bad actor can request retraction of any paper from a retired lab because the originals no longer exist? And along the same lines, how is a trainee supposed to ensure their own protection when they leave the lab and data with it? Those are good questions. Certainly, anybody can post or email saying this paper, published however long ago, from a lab that's still in effect, still working, or is long since gone, looks problematic. So let's say that we get such a thing and the lab is retired, the raw data no longer exists, there's nobody to even really talk to anymore. At that point, the editors are going to be left with a problem. You're probably going to reach out to the institution again. In that sort of scenario, we're probably going to stop with an editorial note of concern. If I can't even talk to the authors and engage them in a satisfactory dialogue, then I'm probably not going to go all the way to a retraction. But it really depends on exactly what we see and what the context is. In the instances when a paper has been retracted due to manipulation, but has scientifically relevant and valuable data later validated by the authors that is important for future knowledge, how can this information still be accessed? Or can it be accessed? The answer to that question is that information can totally still be accessed. The paper does not disappear. The retracted papers at NAR and other journals, they are still there and everything in them is still publicly available. All the data, all the text and everything. All that happens is a retraction notice is attached to the paper with all the problems that imposes on authors. But all the information in the paper is still available. A retraction is just simply the ultimate notice that somebody really clearly did something quite inappropriate to the point where the paper no longer should be viewed in the same manner as an unretracted paper. But the data, the information is still there for anybody to use however they wish. All right, great question, by the way, thank you. All right, so that all is sort of like the big one of data manipulation and image manipulation, of which I've really focused on blots and gels and loading controls, but it can just as easily amount to spreadsheets with large, big data sets that someone investigates and find out was assembled through a computational simulation program instead of actual experimental data. I've seen that sort of thing. And so on. There's lots of examples. Beyond that, you get all the other types of problems that sort of are part of the publication, authorship, review, and editorship. So these are just actual emails over the years that I've seen that I think are interesting. So way back in 2010, today I received this email from an author of a paper I handled. So this came to me from one of the editors who works for my journal. A researcher sent me an email a few days ago inquiring about our method, asking for help with their work related to similar methodology. That person mentioned that someone from blank told him about our method. I don't believe we ever communicated with anyone else at blank unless our manuscript was reviewed there by someone from there. I'm concerned that certain information about our work is being passed around before it is officially published. So hopefully everybody understands. Somebody's submitted a paper, it's not yet published, and then all of a sudden they start getting emails about material in the paper from third parties that shouldn't know about it yet because it's not posted on bioRxiv and it's not published. And indeed, one of the reviewers in this case was actually from redacted, and was the only one of the three that really liked it. So here's a case of a reviewer who certainly looks as if they broke confidentiality and spoke outside the circle of peer review and the editors about the contents of a paper. And in fact, when I approached this reviewer and told them what I had heard, the reviewer actually fessed up fairly quickly and was remanded pretty severely about that, and reminded about reviewer integrity, and told to take a class. All right, and flagged in our journal. Here's another one, 2015. Nucleic Acids Research Advanced Access recently published online a paper entitled blank. I'm listed as an author on that paper with my name misspelled. However, I did not see the draft version before or even after acceptation of the article. So I write to the senior author of that paper and I say, hey, the fifth author listed on this paper, Dr. So-and-so, wrote to us originally to complain their name was misspelled. And then after explaining to them that the spelling of the name was exactly as provided and approved by the corresponding author, that person wrote back to say that, well, actually, I never saw a draft version. I didn't even know it had been submitted. Okay, and so we tell the authors that's troubling. Remember, in terms of authorship integrity, all the authors of any paper should have seen and approved a manuscript before submission and during subsequent revision. So that's a breach of proper authorship protocol. It's not acceptable in the scientific literature. You can't submit a paper with someone listed as an author or really even acknowledged without them seeing the paper. Okay, this is the reply. Dear Dr. Stoddard, I'm extremely upset, but unfortunately not surprised. So-and-so was my former PhD student. She worked under my direction about 1.5 years, initiated the work on the core protein. Unfortunately, during this time, she behaved against the norms of any laboratory, being aggressive and in continuous conflict with almost all students and permanents on the team. Moreover, her work was very poor, generated only limited results that she has been unable to interpret. After extensive mediation, it was unanimously decided to stop her PhD work on our team. She left without getting any PhD degree as well as any significant work. Well, okay, I guess I'll take your word for that, but then why is this person an author, listed as an author on the paper? We hired a new student to restart the work from zero. Her work progressed fast and obtained interesting results that she could consistently interpret. The final paper submitted to NAR contains none of the data from so-and-so. Okay, so again, why is that person an author? Nevertheless, since she started the work, I decided to include her name in the list of co-authors. Knowing her behavior, that was the only way to prevent any complaint from her side. Sorry for breaking the gender confidentiality there at the end. That's my bad, but hopefully nobody's going to figure out who we're talking about here. Well, I understand the difficulties associated with hard conversations and conflict in a lab, but that doesn't by any means excuse an author, certainly a senior author, from following basic proper authorship protocols and matters of authorship integrity, regardless. And we'll come back to that in just another slide or two as I sort of wrap this up. Dear editor, we are taking contact with you regarding a very troubling matter. We just stumbled on the following article recently published at another journal. What is extremely troubling is that this paper was published only a couple days after our own paper finally got published in NAR. However, within this time, the authors managed to perform experiments, compare their results to ours, and even propose a list of recommendations which are astonishingly similar to ours, knowing that their paper was submitted in November and ours was in review since 2014. So they go on to ask, hey, are the authors of this paper by perchance reviewers of our paper, or is this just a really weird coincidence? So I take a look, as a good editor should do, and I write back and I say, well, here's the timing of your paper and their paper. And bottom line is that their initial submission comes in 22 November 2014. Their paper was submitted earlier in that year. Their revised paper came in 5th of March 2015, only 2 weeks after theirs was published online. And it was the revised manuscript where a lot of stuff entered the paper that made citation and discussion based on this published paper right here. So the gap that we're thinking about is from 17 February 2015 to the 2nd of March 2015. So I point out your article was published 2 weeks before their article. It's possible that they saw your article in time to introduce some corrections, some extra text, cite your paper, and so on. It's not out of the question. Then I went back and looked to see and checked the referees of our article and found that none of the authors or colleagues, as far as I could tell, of this article overlap. They're not from the same institution. So I just told them flat out that I don't believe that there was any inappropriate breaking confidentiality. The timeline is awfully short, and I think actually that these people just simply were very, very quick at reading this paper when it appeared online and putting in final bits of text corrections and citations. These authors never accepted that, I will tell you that. We eventually agreed to disagree, and that was that. All right, so those are sort of really, really quickly, just a few vignettes, most concerning data manipulation, but then some about some of these other problems with authorship problems and confidentiality problems. So let's just sort of sum up here with best publishing practices, both from the authorship and the reviewer side. Especially with regards to those final emails. So think for a moment about that email that I got about someone was an author on a paper, and then the senior author, with apparently no guilt whatsoever, said, well, we just put this person on because we didn't want to have an argument. So my advice to all authors, I've already given you my advice about data, but as far as authorship is concerned, and particularly those who are in sort of first author or senior author positions, it just behooves you and is best operating protocol as an author to conduct early, frequent, and repetitive conversations on potential manuscript plans, content, contributions, authorships, and keep written notes and agreements. Try to be transparent, communicate clearly with each and every individual who contribute a reagent, an experiment, or a discussion. So of course, that's kind of hard, right? Because in the early days of any study, right, it's not quite clear how it's all going to shake out in terms of an eventual publication. But at any point in a project, you'll have, at that moment in time, you'll always be saying, well, if we didn't do anything more, this would be the study, this would be the paper we would write. And you should be having conversations early and often about what does our study consist of right now and who's contributed what? And then what do we still want to do to bolster the study, to add data to it, to take it in additional directions, to control for factors that we think are important or are likely to be questioned by reviewers. In my lab, I learned years ago that we just start talking about potential papers really early as is reasonable. As soon as we're getting data that is publishable, maybe not a complete story, but data that in one way or the other is publishable quality data in terms of novelty and in terms of technical accuracy, we're going to start having conversations about what would it fit in with, what sort of a study could this be part of, who would be involved, who are collaborators, multiple investigators in my lab, and we just keep revisiting it. And if you do that, if you make that just a way of life, the conversations start to become very, very straightforward. It's no longer weird. It's just something you do in a lab. And the point is that aside from avoiding misunderstandings or getting problems out in the open quickly, it also helps drive good science because you're talking about the content of the paper and the quality of the data. So I just can't stress enough how important it is to just have really open, frequent conversations about how data might fit into publications. Reproducibility and rigor. You have to look at data and corresponding interpretations, preferably including someone in your lab who's a third party who's not directly involved in the experiment, as it's being generated in real time, not after the fact when the paper's being written. And this is really on the PIs, I think. If you're just letting experiments run in a lab until the point where somebody comes to you and say, we're ready to write a paper, and you're just looking at a lot of the raw data for the first time, you've kind of failed. You should be looking at data generated in your lab in real time on a regular basis. Back in the day when we had all paper notebooks in my lab, I used to just ask everybody, leave your notebooks on the end of your bench. And I come in early in the morning and I would just take a look at the new pages. I just flip through them real quickly, just see what the gels were, see what people were commenting on. And it just allowed me to sort of be in real time, sort of stay on top of what experiments that people are doing. Again, just sort of best operating practice as a scientist and eventually as an author. Same thing, once again, that I've already said, maintaining outstanding records and archiving of experimental data for the professional lifetime of authors and co-authors. Availability and easy retrieval of data, raw data for published figures. And what we always do before submission of a paper, we just make sure for every single image in the paper that we've got the raw data, that I can find it, I can go get it, and I get it all into a folder. That I then keep on my own hard drive, and nowadays I put it in a public depository. So somebody had a good question, which I'm forgetting about maintaining. Oh, it was somebody who said, well, what if I'm an early stage investigator and I'm eventually going to leave the lab? And at that point, I have no control over the raw data. Given that most data these days eventually ends up raw data in some digital form, the days of taping gels and X-ray films from blocks into a paper notebook are long gone. You should have access to the raw data, and you should be able to download and store it all on your own hard drive, or at least make sure that you guarantee that that data is maintained in an electronic form. And again, you're only worried about data corresponding to published figures. And published figures should be backed up in this day and age by archived raw data. So it shouldn't really be a problem anymore for leaving the lab. Back in the days when it was notebooks that were in file cabinets and stuff taped into notebooks, you're absolutely right. Those things would eventually disappear. But these days, moving forward with all of you at your stage, this stage of training, shouldn't be an issue. And it should be something that just is taken care of one way or the other. And it should be part of your exit from a lab, is where's all the raw data and how is it being accounted for and safely stored for the rest of my career. Multiple authors, starting and ending with a senior author, have to examine the relationship between raw data and published data. Again, both in real time and certainly immediately before submission. That's your last chance to go through all the data and take a look at the underlying data, make sure it's ready to archive, make sure it's all in good shape. As far as your writing, copy and paste is the enemy of original unimpeachable writing. Most journals now use plagiarism checkers. Most journals are not understanding of copied text, even from your own previous work, even methods. Best to write clean text, regardless of how many times one has described background literature, a method. I will say that, obviously it's a pain to rewrite like a protocol that you use over and over again. These days, my solution to that problem is I will always, in my methods, just cite and summarize a protocol. And then in supplementary methods, I will quote the protocol with quote marks around both sides exactly and directly, and I will recite the previous paper just to make sure that I can't be dinged for self-plagiarism or any other form of trademark infringement with regards to written text. As an author, identify and disclose any and all potential conflicts of interest, intellectual property, involvement with companies that may make use of any of the data, any financial connections you have to any form of the data, anything else, connections to foreign, research organizations that are within foreign countries is a huge one. Just be very open and honest with potential conflicts of interest as an owner, and err on the side of over describing and over disclosing. Nobody ever got in trouble for disclosing trivial things. Okay, reviewers, and I think this is the very end of the talk, right on time. So this all goes hand in hand, right? Good authorship practice should be coupled with good reviewing practice. So identify and disclose any potential conflict of interest. So if you get a request to review a paper from an editor, if you have past connections with a researcher, maybe only talking about research, maybe an old collaboration that was never published, just disclose it. Just send an email, say, hey, I'd be happy to review this paper, but I just want to disclose that I've got the following connection with author A or author B. What will always happen is the editor will thank you. The editor will say, hey, that really is not a problem. I have confidence in your ability to review this paper fairly and in a rigorous manner, but I will go ahead and add a note that you disclosed this. So you're protected as a reviewer as soon as you do that. So err on the side of over disclosing. Nobody ever got in trouble for asking about, I had a conversation at a conference 15 years ago with a middle author on the paper. The editor will just thank you, say that's fine, and then they'll enter it into the record. Maintain confidentiality very carefully. You don't go talking about any aspect of a paper with anybody outside of yourself without getting prior permission. And that includes if you want to include a colleague in your lab, or if you're a PI, if you want to have a graduate student or a postdoc participate in the review, you have to go back to the editor and tell them and ask for permission. You'll always get permission, and it will get noted again. And but you never ever break confidentiality in any form. You don't tell someone over lunch about a cool paper on your desk that you're reviewing. You just don't. Because if somebody tells a friend to somebody who tells a friend to somebody about something cool they heard, and it gets back to the original authors, it will bounce back to you as the reviewer. Remember that I can always go and see who reviewed any paper. So if I get an email from an author and saying, I can find out who the reviewer was, and it's never good. Kelsey says, should a reviewer also disclose a clinical relationship with an author or only research relationships? I'm not sure exactly what that means. And may I leave now, please? Yes, we are over. So, and I'm on my last slide. So, Kelsey, I don't quite understand the question. So I'm going to ask you, if you want to follow up on that, please feel free to drop me an email. And I'd be glad to explain what you mean, and I'll be glad to give you my opinion for sure. I just don't have a good answer off the top of my head. All right. If you recognize a conflict of interest or inappropriate overlap after accepting a review request, recuse yourself, explain the situation, and never, ever, ever contact the author of a paper until it is in the public domain. And again, go to COPE, publicationethics.org, and read their ethical guidelines for peer reviewers. And so that's my last point. Is the other point that I would make, aside from raw data is everything, and good conversations in a lab about authorship are everything, is go to the COPE website and spend some time, a couple hours at least, sort of like taking a look at their basic ethical guidelines for authors and reviewers. And with that, I think we are done. That is my last slide. So sorry for running a couple minutes over, but there were some good questions that got asked. I'm happy to stay online as long as any attendees want to ask any follow-up questions. I'm not going anywhere, but I do know people have places to be.
All right, thank you so much, Barry. That was a fascinating talk, and I really appreciate you sharing your responsibilities and actions as an editor, and great advice about archiving your data and best publishing practices, and especially warnings about what to do if you as a scientist are being questioned by the internet. I really appreciate your sharing.
Yeah, it's a brave new world out there with regards to social media. Like everything can put an enormous amount of pain on a person. Cancel culture, regardless of what you think of it, extends to scientists just as much as any other walk of life. And so just it's just so incredibly important to really, dot your I's, cross your T's, and have that raw data.
Absolutely. It looks like all the comments are just thank yous, very informative, super helpful.
I see them.
So thank you again, Barry, and I think we can probably conclude.
All right. Well, thank you so much for the opportunity. It's a pleasure as always.
Thank you. Take care, Barry.